decision to employ advanced systems for AML modeling was made by our clients, and the need for a co-clinical capability of taking liquid tumors and rapidly forming mouse models with the human patient sample. And so the technology was piloted in academic circles dating back to 2007, so it's relatively new technology, and it required the use of advanced mouse systems, and our medical affairs team was a decided advantage that we could be literally in the clinic and recruit patients, and we were successful. We have two platforms, and one is ideal for modeling large numbers of mice from single patients, and so this is called leukapheresis samples. And leukapheresis is a rarely done emergency procedure, usually with AML patients, where their AML blasts are removed from their blood. And this is an excellent source of leukemic stem cells from which we can make mouse models of the patient. And so from one leukapheresis sample, we could literally get enough AML cells to create 200,000 mice on study, and we generally purify and archive enough cells to do 10,000 mice per patient. And so today we have over 40 models established with our clinical collaborators. They're characterized for their ability to make models in the mice. They're characterized for genomic mutations, and in some cases for drug response. And collectively, they give a nice representation of the patient population, because importantly, AML is one of the most heterogeneous diseases, and it's also a very difficult disease to model, and it's almost a labor of love, and we have had great success with this program. Leukapheresis is rarely performed clinically. Maybe a large clinical site will do this procedure only a couple dozen times a year. So if you needed a particular phenotype or genotype of a patient profile, it would be impractical to rely on leukapheresis models. You just wouldn't have enough random patients coming through the door. And so we started examining how can we model at will any patient as they progress in clinic. And so what is the most practical way is to use low volume blood draws, peripheral blood draws. And so a 14 mil blood draw as the patients progress in clinic. And so this allows us, with our medical affairs team, to literally dial in what kind of patient we want to model and simulate a clinical trial. And we've learned how, from one simple low volume blood draw, we've learned how to make about 200 mice on study, which is a really large cohort. And so low volume blood draws allows you to model co-clinically patients as they progress in clinic in a non-invasive way that generally has 100% patient compliance, physician compliance, doesn't require dramatic IRB alterations, and allows us to model AML cohorts of patients of any demographic. And so it's a very exciting advance, and it's a great tool for translational studies, but also co-clinical studies. And we have a large program, phase 1 through phase 3, where we're literally modeling our sponsors' patients direct off of their clinical trial. We have, in addition to AML, we have the capability to model ALL, and we have the ability, we've been collecting ALL samples from both T-cell and B-cell ALL patients through leukapheresis, but in all practicality, this could be expanded to the co-clinical low volume blood draw dial-a-patient system, if you will, that I mentioned. And we're working on research programs that will manifest themselves next year, where we're exploring other diseases such as CLL and multiple myeloma, which have heretofore been very difficult to model co-clinically. So we see a great future for Hem-Onc and an increase of the therapeutic areas.